right, our next session includes two speakers. The first speaker is Scott Coffin. Dr. Coffin is a research scientist at the California State Water Resources Control Board. And following Dr. Coffin, we will hear from Yasir Sultan. Dr. Sultan is the acting director for the Water and Air Quality Bureau at Health Canada. So over to you, Dr. Coffin.
Thank you, Robin. Just going to pull up my slides. Alrighty, everybody can see my slides. So in this presentation, I'll provide a summary of the research and actions conducted by the California State Water Resources Control Board, and as well as our partners to address microplastics impacts to humans. First, a disclaimer: the views expressed here are solely those of the author and do not necessarily represent the views or policies of the California State Water Resources Control Board or other state agencies. Findings of microplastics in tap water in 2017 prompted concern in the California State Legislature, which led to the passing of Senate Bill 1422 in 2018. This bill requires that the State Water Board first define microplastics by July 1st of last year. And by July 1st of this year, we're required to adopt a standardized method for monitoring, provide accreditation for laboratories, conduct 4 years of monitoring and analysis in California's drinking water, and consider adopting a health-based guidance level so that when we inform consumers about contamination in drinking water, we can tell them the findings of the state of the science. So first, starting with the definition, microplastics are defined by the California definition as being solid polymeric materials to which chemical additives or other substances may have been added, which are particles which have at least 3 dimensions greater than 1 nanometer and less than 5,000 micrometers. Polymers that are derived in nature that have not been chemically modified other than by hydrolysis are excluded. So this is a rather broad definition that's based off of harmonized definitions from the scientific community, and we believe is inclusive of the particles that are of concern for human health. So now that we've defined the class of contaminants, we can develop standardized methods to monitor for them and accredit laboratories so that we can be confident with those results. Prior to the passing of Senate Bill 1422, no standardized methods were available, so we had to develop our own. Standardizing methodologies for monitoring a contaminant is a first and critical step to regulating a pollutant. Through a partnership with the Southern California Coastal Water Research Project, or SCWRB, we developed standardized methods using 2 types of instrumentation, infrared and Raman spectroscopy. Methods were evaluated through an inter-laboratory validation study that involved 26 participating laboratories from 7 countries, representing academia, industry, as well as government. The standardized methods are available on the State Water Board website and are written in US EPA method format. So to assess the method performance, we first created a set of blind samples that are representative of the complex mixtures of particles that we find in drinking water. This includes 4 different shapes and 4 different polymers of microplastics, ranging in sizes from 1 to 1,000 microns. We also included false positive materials that can be easily mistaken for microplastic particles. And this blind sample was developed specially for this project from the Norwegian Institute of Water Research. We then sent these blind samples to laboratories who extracted them in the lab and identified each particle and categorized them by their shape and their size, as well as analyzed the chemical profile of these particles to determine whether or not they're plastic. Based on the data received from these laboratories, we were able to assess the accuracy and precision of each of the methods. So first, I want to focus on the overall findings of the spectroscopic methods for identifying the particles in the mixtures. On the y-axis here is the percent recovery and a dotted line showing where the 100% mark is. The closer the measurements are to 100%, the more accurate the measurements. And for overall total particle count, we can see that laboratories had relatively low recovery, but this is not the whole answer. If we break the answer up by size fraction, we can see that laboratories had a challenging time identifying the particles smaller than 20 microns, with only 4 labs even attempting to detect these particles. But when we look at the larger particles, 20 microns and above, we now see that laboratories can accurately report the number of particles in a fairly precise way. We also see some differences between the colors and the morphologies that are reported, with some particles being reported more often than they should have been, and some particles being reported less often than they should have been. Overall, looking at the method's strengths and weaknesses, we feel that we have two methods that are ready to use for monitoring in drinking water, which are Raman and infrared spectroscopy. These two methods are highly accurate for identifying plastics, with accuracies ranging from 83 to 93%. And the biggest difference between these two methods is that Raman takes a little bit longer and can be a little bit more expensive, both for the instrument and for the consumables cost. There is a strength to using Raman spectroscopy over infrared, which is that Raman can get smaller particles down to 2 microns or so. So Senate Bill 1422 requires the State Water Board consider adopting a health-based guidance level so that we can inform consumers about what we know of the human health effects of microplastics. As we learned from Dr. Ellis Hutchings, the human health impacts of microplastics are not well understood. We partnered again with the Southern California Coastal Water Research Project to facilitate an expert workshop to critically examine the microplastics toxicity literature. We examined literature for both drinking water and for ecosystem health effects. And in this talk, I'm only going to be talking about the human health impacts. But if you'd like to learn about our ecosystem risk assessment, you can find videos of recorded presentations on our website. So similar to the approach employed by Dr. Ellis Hutchings, we screened microplastics laboratory toxicity studies for their reliability and relevance using a set of quality criteria corresponding to experimental design, risk assessment applicability, and particle characterization. Starting with 66 mammalian toxicity studies, 31 of those used in vivo animals, which we prioritized for screening and further evaluation. Of those studies, 12 were deemed fit for purpose that we went on to further evaluation with expert review. And it should be noted that that none of the studies met all of the quality criteria. So we had to use a more lenient set of quality criteria to be able to examine any of these studies through expert review. So starting with the overall findings, of the 12 studies that were deemed fit for purpose, the subject matter experts concluded that 7 of these studies contained reliable toxicity information. Of these, 4 studies reported endpoints on male reproduction and 2 reported endpoints on female reproduction. Studies consistently showed adverse impacts to sperm deformity and count, as well as decreased testosterone in rodents. All of the studies reporting adverse impacts in rodents used particles that are 20 microns or smaller. Dose response of critical effects were modeled using EPA's benchmark dose modeling software. The point of departures for each critical effect is shown here, and we can see that there are a relatively tight agreement between the impacts, between the concentrations where we see threshold effects for male reproductive impacts, such as sperm count, testosterone concentration, sperm motility. And the most sensitive impact that we observed here was for decreased anti-Mullerian hormone concentration, which is a biomarker of impacts to female reproduction. So despite these findings, there's some critical data gaps that prevent us from reliably assessing risks or deriving regulatory levels for the protection of human health in drinking water or otherwise. First off, the effects database is largely inadequate for assessing the exposure or the hazard of all microplastics. Most of these studies used poor particle characterization, and they didn't report a lot of information that we deem critical. They also used a limited set of polymers, shapes, and sizes, with almost all of the studies using polystyrene spheres. Additionally, we have uncertainties with regards to the effect mechanisms of how these particles are causing toxicity, with some evidence pointing to inflammation and oxidative stress. However, this is without this understanding, we can't extrapolate the effects found in polystyrene spheres to the diverse particle types that we're all exposed to. Finally, we have an incomplete exposure database with very limited information about what we're eating through our food and no harmonized drinking water data to date. We can partially fulfill this last requirement through our standardized method and our monitoring campaign here in California. So luckily for us, as Dr. Ellis Hutchings mentioned, the field is rapidly progressing, and we're seeing an exponential increase in the number of mammalian toxicity studies for microplastics. We recognize that the studies are coming in fast, and we want a way to reduce the workload for further risk assessments and meta reviews. So we built an interactive data visualization app called Tomex. This is a web-based platform that is free and open to the public, where you can look at the data that we assessed through our expert workshop. There's a lot of statistical tools on here, as well as visualization tools. And this is also a living database. So anyone that publishes a peer-reviewed study on microplastics impacts to humans or ecosystems can upload their data directly to this database. Again, for the purpose of making more rapid assessments of toxicity impacts in the field. Based on our research on the health effects and analytical methods, we've developed a plan for 4 years of testing and analysis of microplastics in drinking water. Since no government in the world has ever required monitoring for microplastics in drinking water, this will be a trailblazing effort for the state of California. So in designing a monitoring campaign, it's important to first understand where we would expect to find microplastics and look there first. First off, we know that microplastics are unlikely to occur in groundwater and are likely to be found in surface waters that are used as drinking water. We also see that treatment can reduce the concentrations of microplastics, and I'll talk about that a little bit more later. As far as bottled water goes, this is often given as a replacement for contaminated water to people in California. But in the case of microplastics, we probably would not suggest this, as we understand that the plastic packaging itself can contribute microplastics, which is one of the reasons that we're finding on average about 3 times higher concentrations of microplastics in bottled water than in tap water. So microplastics removal in drinking water is size dependent, as shown here. And as a reminder, 20 microns is our lower size limit for quantifying microplastics, which is approximately about the size of bacteria. A lot of this is going to be removed through most treatment processes. So this is an overall timeline for monitoring in drinking water in California. We're going to start by looking in the source waters in 2022. And for after 2 years, we hope to have advanced methods, at which point we'll be able to look for microplastics in the treated waters, looking at smaller particles. So there's multiple pathways for microplastics impacts to humans, exposure to humans. And I want to just cover really briefly an additional pathway going through the food chain. So as plastics of all varieties enter the wastewater treatment plant, they can be discharged into water bodies, and they can also be land applied onto agricultural lands, which there is a potential for human exposure. So wastewater treatment plants are generally effective at removing microplastics, with 88 to 99% of microplastics entering the sludge. A recent study estimated that approximately 1 quadrillion microplastics are land applied in the United States on a yearly basis through this application. After processing, a portion of sewage sludge is converted into biosolids, which are nutrient-rich organic solids that are often used on agricultural land. And we've seen, looking at the content of microplastics and biosolids from 1950, a strong positive correlation to the amount of global plastics that are produced. At this point, we don't know if this is actually causing impacts for human exposure. However, we do have reports of microplastics impacting the growth of plants, as well as the reduction of fruit produced by these plants. So this is something that we are considering doing some monitoring for to further examine the effects. And just want to mention really briefly here about bioplastics as a replacement. Not all bioplastics are created equally. Some of them degrade rapidly. Some of them can take very long time to degrade in the environment. And when considering replacing plastic with bioplastics, it's important to consider the biogeochemical flows and the limited quantities of phosphorus and nitrogen in the environment, whether or not we should be growing plastic with these limited nutrients or food is a decision that we'll need to encounter soon. Additionally, I just want to leave you with one last thing. Our Safer Consumer Products program for the California Department of Toxic Substances Control is considering regrettable substitutions to plastic, such as bioplastics, as we move towards trying to reduce the total plastic that we consume and that we're exposed to. So some upcoming policy and regulatory actions in California. We will begin monitoring in drinking water and doing some environmental monitoring starting next year. And we are additionally validating these methods for additional matrices. So we have these standardized methods for drinking water, but we're also validating it for sediment, fish tissue, and ocean water as of next year. And as mentioned before, the Safer Consumer Products is evaluating microplastics impacts to human health and the environment to consider better alternatives. Thank you very much, and I look forward to answering your questions.
Thank you, Dr. Coffin. In the interest of time, we're going to actually have Dr. Sultan speak first and then have a joint Q&A period, if that works. So over to you, Dr. Sultan.
Hi, and good morning. I hope everyone can see my presentation. Thank you very much, Robin, Heather, and the planning committee for thinking about me, inviting me to this very interesting webinar so far. I've thoroughly enjoyed the presentations, and I will be spending some time, I'm just gonna mute this here. Spending some time talking about the Canadian activities related to both macroplastic and microplastic pollution. Here in Canada, we have taken sort of a upstream and downstream approach to plastic pollution to look at macroplastics as a whole and understand that their sources are microplastics, and taking a look at microplastics as well. Talk to you briefly about the Canadian context. Our plastic science agenda, we've been working on since 2019. Our recent science assessment of plastic pollution, similar to the work done presented by others, we have undertaken a review as well on plastic pollution. Some ongoing research, as mentioned, it's the microplastic research is very much an emerging area. We have people both from the world of particles and traditional chemicals who have been who are now looking at microplastics. So we're starting to see that increase in papers coming out, and our understanding seems to improve overnight as time goes on. And some of our regulatory actions that we are proposing to take here to reduce plastic pollution and with some next steps. So for us, we've been looking at plastic pollution for a very long time, as has every other country in the world. For us, a coherent strategy emerged in around 2018 around the G7, G8 discussions, where we launched the Ocean Plastics Charter to start to look at macro and microplastics in the environment. We then, with the Canadian Council of the Ministers of the Environment, which is our provincial territorial partners, we worked with them to set up a zero plastics waste strategy to move us incrementally towards zero plastic waste till about 2030. And then we launched the Canada's Plastic Science Agenda, which is very much, I'll get into the following slides. And as part of that whole body of work, we did conduct a science assessment of plastic pollution in 2020. And we published a discussion paper on the integrated management approach to start to see, are there any reductions we can make of certain type of plastics to get us towards reducing our footprint of macro and microplastics in the environment and reduce harms to humans and Canadians. So the plastic science agenda is a pragmatic approach, as I mentioned, upstream and downstream to dealing with plastic waste. It's a multidisciplinary view. It's got 5 different themes that are underpinned by collaboration, knowledge mobilization, capacity building, and a circular economy lens. We don't want to just deal with plastics. We want to make sure that the end of life is dealt properly. They're getting back into our economy and keep keep contributing to their value-added uses. So where we have focused very much is on the first two themes here, is on the detection, quantification, and characterization of plastics in the environment. As you saw from the previous presentations, studying and detecting and quantifying microplastics is very difficult. We're used to, in the particle world, we're used to, adding some kind of marker to particles, be it manufactured nanomaterials or certain type of fibers, and then studying them and understanding them. But plastics are a little different. They don't necessarily stand out in any way, like something like metal oxides do. So we use generic techniques like Raman spectroscopy or FTIR and others to try and build some footprint information, which makes it complicated because there's so much other type of materials in the soup that we study. So we're also trying to build capacity understanding on impacts on wildlife, human health, and the environment. I won't dwell on that. It's been discussed in the previous presentations. And to complete our narrative, we are also looking at better understanding plastic design and alternatives, such as what was presented on the bioplastics in the previous presentation, sustainable use of plastics, and waste diversion and recovery of plastics here. So understanding the potential health of microplastics, we have a multifaceted approach here. Domestically, the first one is we did a science assessment of plastic pollution, which looked at macro, so above 5 millimeters, and micro, under 5 millimeters. To summarize the current state of the science, it was published last year regarding the potential impacts of plastic pollution on the environment and human health. Looking at the presentations that have come before me today, it's clear that the science is evolving so rapidly. Even 10 months when we published the science assessment is, I would say, not completely up to date anymore. So that's just evidence of how quickly the field is evolving. We're also guiding future research and ongoing research here to improve our understanding of microplastics and macroplastics as well. And we are fostering Canadian capacity both on research within Health Canada and external in our academia. And I'll present some of the work that is ongoing. And we're hoping all of this informs our decision-making on plastic pollution Canada and gets us towards that zero plastic waste ambitions that our government has supported. So we undertook a science assessment of what we call a fairly thorough science assessment, the first one of its kind to bring together both eco and human health impacts together into one document, back, at least in 2020. What it found was that, as you've seen, there are many sources that contribute to plastic pollution. Microplastic, what we concluded is macroplastic pollution is harmful to the environment and wildlife. So these are larger chunks. We saw some pictures of the birds ingesting these larger chunks. And then, when you think about the garbage patch and some of the microplastics that are found there. We conclude that information on the impacts of microplastics on human health and the environment is limited. This is consistent with the theme that we've heard today. We're just starting to scratch the surface. The first thing, of course, is we need the methods. Then we need to understand real life exposures. Then we need to understand hazards. And then we need to make sure we're using the right metrics to measure some of these things. So the field back when we published the assessment was still limited, still is. We also concluded that research is needed to advance the understanding of the impacts of microplastic pollution on the environment and human health. I'll talk about some of those themes that we identified, and action is recommended to reduce macro and microplastics that end up in the environment. So in terms of the findings on human health, specifically for macroplastics, so the larger chunks, we found that it's not anticipated to be a concern for humans. There are some potential effects related to physical impacts from the chemicals present in or on the plastics, be it macro or micro. As I mentioned, there's limited information on human health effects of microplastics, and there, as you saw earlier, there is no clear answer because the studies are not all comparable and there are no standardized methods. So there's a divergent view in lab studies. So some studies report adverse effects while others don't. And it's very difficult, or it's really not possible right now to link those to the general population because of the level of uncertainties that we have. And just to close on the assessment findings, there are also reviews. We noted that there are reviews by the WHO, EFSA, and the FAO that indicated a low concern for human health from chemicals present on microplastics from food or water. So in terms of what we did find, current data on the occurrence of microplastics in food is limited, as you've seen today. Most of the information that's available on findings is in seafood, because you get to see some of that trophic transfer. We have seen microplastics detected in some bottled water samples and as well in tap water. So there is that exposure that's happening. We identified a few research themes that need to further develop. The first one is standardized methods, which I think is perhaps the most important need to properly move us forward in this field of microplastics. And we would like to start, there are a lot of studies underway, as you saw in California and others, looking at Europe, looking at some standardized methods. We need to further the understanding of human exposures to microplastics, including understanding sources. And then there's further the understanding of ecotox effects. When we looked at the monitoring efforts underway, we found very little comparability across different monitoring efforts. So we need to improve the level of consistency in monitoring for microplastics, not only in water, but also soil and air and other environments. We have some internal research we have started within Health Canada. The first one is detecting and measuring microplastics in drinking water sources. Show some data in the next slide. Understanding that microplastics can get airborne, we're looking at developing methodologies and collect samples in indoor environments for airborne microplastics to look at the inhalation exposure route, and detecting and characterization microplastics, including nanoplastics in foods. We also have some health effects work underway as well, detecting microplastics in tissue following oral exposure, detecting and characterizing microplastics in human placentas, and studying the interactions of microplastics in cells. Many of these studies are just started within the last year or just starting now, so it's a bit early to provide any data on this work. The one study that we did start a little early in our process with some information is our work on testing Canadian drinking water for microplastics. The pilot study, it tested the intake, post-treatment, and within distribution system from 5 drinking water treatment plants. We wanted to understand how each step of the treatment process led to changes in the microplastic concentration, microplastic type, and quantity. This was work led out of McGill University, and what we found was that removal efficiency during drinking water treatment was above 90% of particles above 20 microns. But it was also, given that we didn't have any standardized methodologies, made it very difficult for us to do this study and do it properly. So it took some time. We also have other ongoing work within Health Canada. Here, we are looking at microplastics in new homes. So we're collecting samples from settled dust to better get some baseline information if there are microplastics there, and then look at it, in some older homes as well. We're also developing methods for ambient air particulate sampling and quantification for microplastics less than 2 microns, and fine particulate air samples as well. This also did indicate that presence of a variety of microplastics are in the respirable size fraction, so 0.1 to 1 micron. In addition, we're working on a test system for the exposure of lung cells to microplastics under conditions that model real-life human exposures. So getting back to that uncertainty around how to link lab studies to the general population. This will enable testing of the airborne contaminants for microplastics at realistic concentrations using human relevant models. We also have supported external work to build capacity in our academic institutions here. One is under the Plastic Science for a Cleaner Future program with the INRS in Quebec, assessing the toxicity and inflammatory response associated with exposure to nanoplastics. And under the Increasing Knowledge of Plastic Pollution call for research, one project with U of T, University of Toronto, on the occurrence of microplastics in drinking water and impacts on biofilms in drinking water infrastructure, and one with Ryerson University in Toronto, Degradation and Transformation of Plastics in Water and Impact on Mammalian Cells. So just to close up our research side, I would say that we're still emerging very much in the realm of building capacity in both internal and external. And it's going to take us, I would say, a few years still to develop some comparable, reliable data that we can use for decision making. Following the science assessment, I want to just close on our regulatory action to date. Based on the recommendations of the science assessment in 2020, we published a discussion paper to consult Canadians and stakeholders on options to address plastic pollution. The paper proposed banning or restricting certain single-use plastic items like checkout bags, straws, cutlery, stir sticks, six-pack rings, and food serviceware made from problematic plastics, creating recycled content requirements, and ensuring end-of-life responsibility. So in May 2021, we took the first step towards that. We published plastic manufactured items to our Schedule 1 of the Canadian Environmental Protection Act, which is an administrative tool to signal that we would like to start looking at some risk management action. Follow-up has not been published yet. So in closing, we're continuing our domestic engagement to implement Canada's zero plastic waste agenda. So the ambitious goal to have zero plastic waste by 2030. And part of that is to continue the scientific leadership, both domestically and internationally, on human health and human health and environment, and to reduce plastic pollution. And in that vein, we are continuing to increase our capacity here domestically and leveraging internationally to address data gaps. And that's it from me. Thank you very much.
Thanks very much. And I will invite, there we have, Dr. Coffin is back as well. All right, so we had a number of questions, but I'm going to start with one question for each of you. So Yasser, the first one is for you. From a dietary perspective, the food, digestive, and feces, together with the physiological cellular functions of the gastrointestinal tract, are important considerations in assessing exposure and hazard. What are your thoughts on this?
That's a great question. And of course, it goes to the uncertainty that exists in the science right now. We would like to better understand how the digestive tract is interacting with the microplastic. What are the changes? Are there other chemicals leaching off with it? All of these we don't have answers to. So of course, we need more answers to the questions. We need more science. And as I mentioned, that we are, I would say, still a few years away from answering that question. Of course, it's plausible theory, but to be tested in the lab, we will need more studies.
Thank you very much. And Dr. Coffin? It starts out with a compliment. So good to learn California developed a 4-year plan to monitor microplastics in drinking water. Can you elaborate, though, on why microplastics are not expected to be found in groundwater sources?
So of these studies that have looked for microplastics in groundwater, almost none of those studies have found detectable levels. And the reason for the unlikelihood for ever finding microplastics in groundwater at significant quantities is that the groundwater matrix, the particles in the sediment, are likely doing a good job at capturing those particles. And there's a caveat here. There have been a few studies that have looked at gravel as an infiltration bed for groundwater. There are some water plants that recharge their groundwater using highly mobile media, such as gravel. And under those circumstances, we are seeing contamination of groundwater by very small microplastics, so 1 micron and smaller, which are very challenging to detect. We can't currently quantify those particles with our Raman and IR methods. So it's not to say that we won't ever find microplastics in groundwater at significant levels, but current reports suggest that there are very low levels of contamination.
All right. So I think I'll leave it there until we're back with the panel. And thank you both